Next chapter, Bioremediation, Introduction and Concept. Industrialization is a necessary evil. Nowadays, the rate of industrialization is going on increasing. Lot of industries are established in our area. However, the rapid industrialization revolution creates the pollution. It is a necessary evil means it is necessary, but sometimes it creates the problems also, or sometimes it creates the disaster also. The problems in the form of the pollution, it creates the pollution. Unless and until the pollution is less, unless and until the attempts that has not been applied to reduce the pollution, the rate of pollution that will be increased and which will create the problems in the human health and hygiene also, as well as in the vegetation, as well as in the environment also. That's why the rapid industrialization that creates the pollutions and the various toxic gases, industrial effluents released from the industry to lead the prolonged pollution of the habitats. For this, bioremediation, it is a remedial activity that has been applied to reduce the rate of pollution, to minimize the rate of pollution as such. And the bioremediation, it is the use of living organism to degrade the environmental pollution or to prevent the pollution. It means that the biological tools that can be utilized for the purpose of to minimize the rate of pollution to such an extent to prevent the pollution as such. The aim of bioremediation is to complete the mineralization of organic pollutant to carbon dioxide and it release on stimulating the growth of microbes. The main goal of the entire bioremediations, or the main aim of the bioremediation, is to complete the mineralizations of the organic pollutants. Now we know that the organic pollutant that has been directly liberated from the particular type of the industry, and that effluents or that pollutant that create the problems to in the public hygienes, as well as they enter that habitat of the particular type of the plants also. And that is why sometimes the water is get polluted as well as the soil is get polluted. That water and that soil, when it is polluted by the help of the hazardous chemicals, when it get polluted by the help of the toxic elements like the cadmium, like carbon monoxide that is in the air pollution, the cadmium, lithium, arsenic, such type of the pollutant that has been directly liberated from the particular industry, and it is in the form of the effluents. And that effluent, if it is not well disposed, or if it is not well treated, it will if it is directly sent to the enter that water bodies, or if it is directly sent to the enter that soil, the soil is get polluted, the soil become barren, water it get polluted, it is unfit for drinking purpose. Not only drinking purpose, but it is also unfit for the agricultural purpose. For that purpose, the bioremediation is utilized, and this bioremediation is nothing but the complete mineralizations of the toxic elements into carbon dioxide, and it releases the stimulating of the growth of microbes. It releases whatever they enter the carbon dioxide and released carbon dioxide that will be absorbed by the help of that growth microbes for the growth and the development of that microbe as such. That uses that microbe that uses the contaminants like oil, solvent, pesticides, etc. as a source of food and energy. The types of bioremediation. There are two types of bioremediation. in situ bioremediations and ex situ bioremediations. In situ bioremediation, it is applied to eliminate the pollutant in contaminated soil and groundwater at the site of pollution. It means that the in situ bioremediation, that is nothing but the whatever they enter the pollutant which are present as it is on the groundwater or on the entered that soil, the particular type of the entered that policies that has been applied or the parameter that has been applied to remediate that groundwater as well as that soil, that is called as the in situ bioremediations. These techniques are less expensive because saving of transportation of the cost and using of harmless microorganisms to eliminate the chemical contaminants like chlorinated solvent, dyes, heavy metals, as well as the hydrocarbon pollutants and the polluted at the polluted site and at the polluted site. In situ bioremediation, it is carried out by following method. First, bioventing. Venting, it directly states that there is a sort of the ventilations, and that ventilation that is applied or ventilation, it is facilitated with the help of air. In this technique, the air and the nutrients are supplied aerobically to contaminated soil so that the indigenous microbes are get stimulated and degrade the component. So in the bioventing, the particular air is passed, and that air is passed due to which some that microbes which are present within that soil or whatever the microbes which are present within that enter that groundwater or enter the water body which are in the form of the river also, if that water of that river is polluted by the help of the any type of the heavy metals or any type of the toxic or hazardous chemicals as such. These these microbes, these microbes are get stimulated. They get stimulates, and that stimulation that tends to form or that leads to degrade the compound, whatever the chemical compound or whatever they enter that toxic compounds are present within that soil or within that water body. That's such concern. The air that is oxygen is supplied through direct injection by means of well. The well is done. Well is some water, so-called as the soil is excavated, and that excavated soil that forms the well, and by the help of that well, the air is passed, and that well it is used to site polluted with the spilled petroleum product. Whatever the petroleum products are there, that petroleum product that are directly in the form of the spill, and by the help of that air, that petroleum product are bioremediated. Second, that is the bioslurping. Bioslurping, it is used by providing the oxygen indirectly to remediate the soil contaminated by the fuel and the groundwater contaminated by the light non-aqueous phase liquid, that is LNAPL. It is also used to remediate the soil contaminated with the volatile and semi-volatile organic compound. Whatever the volatile compounds are there, that can directly evaporate, or some semi-volatile components are there that can be evaporated or semi-evaporated, that can be directly bioremediated or that can be directly removed by the help of the microbial activity. These microbial activity that starts due to the injections of the particular type of the enter that air, that is in the form of the aerobically, and these aerobically air that has been directly supplied, so that there is some stimulation that has been directly formed, a stimulation that can directly forms to the some microbes, and that microbe that can directly take or absorb that toxic or hazardous chemicals. This system uses the slurp that extend into the free product layer and draw off the free product and the soil gases. The pumping lifts the LNAPL, that is the liquid, that is nothing but the liquid, non-aqueous phase liquid, to the surface where it's separated from the water and air. Next, that is the biosparging. In this method, the air is injected into the soil surface at saturated zone to stimulate the microbial activity in order to promote the removal of the pollutant from the enter that site. In this method, the air is injected into the soil. The soil is get injected by the help of the air surface, and whatever they enter the saturated zones of the particular type of the toxic chemicals are there. When the toxic chemicals, if suppose that area is suffered by the help of the heavy metals, heavy toxic chemical, heavy hazardous chemicals, and that heavy hazardous chemicals or toxic metals are that are stimulated, that stimulate the microbial activity. Due to that injections of the air that stimulate the microbial activity, and that microbial activity that promote the removal of the pollutant from that area. This is nothing but the biospurging, so that there is upward movement of the volatile organic compound to unsaturated zone to promote the biodegradation, and it is then biodegraded. That role of the enter that microorganisms, or role of the enter that microbial activity, that is nothing but to degrade that toxic chemical, that hazardous chemical, or the heavy metals. This technique is widely used at site contaminated with petroleum product, especially diesel and the kerosene. Second, that is the ex situ bioremediation. This technique involves the excavating pollutant from the polluted site and transported them to the another site for the treatment. This is the expensive method, you may say. As the in situ bioremediation, that is a less expensive, that is not a costly. But here, that is the ex situ bioremediation, that is nothing but whatever the area is get affected by the help of the pollutant, whatever the area is affected by the help of the enter the effluent that has been directly released from the industry, that area, that material or that zone is directly excavated, and that excavated quantity of the enter that area, that is directly transported to the another area where the particular treatment is given to them. And this is the expensive method, that is the ex situ bioremediation as such concerned. They are as follow. First, that is the biopilling. Biopilling means the pills are formed, and these pills are formed due to the enter that microorganism activity or the microbial activity. In this method, the excavated soils are mixed with the soil amendment placed on the treatment area and the bioremediated with the forced aeration, either by supplying the air under the positive pressure or draw the air with the help of the negative pressure. It means that excavated site where the material is taken and it is transported to the another area where the treatment is given, and in this treatment the air is supplied to that excavated material that has been taken out from that polluted zone area, or they enter that area which are having the large number of the pollutant. That material, it is then directly supplied under the positive pressure of the air or negative pressure of the air. It means that the air is forced, air is passed forcefully, or air is taken out by the negative pressure. And this negative pressure that can start the negative and the positive pressure, that can stimulate or that can directly facilitate the microbial activity. And this microbial activity, it is enhanced through the microbial respiration, and the biopills are used to reduce the concentration of the petroleum pollutants in the excavated soil. Now second, that is the windrows. The windrows relay, that is depend on the periodic turning of the peeled polluted soil. Whatever the polluted soil is peeled in the form of the peels, that polluted soil mixed with the water and bring about the increase in the aeration. It is mixed with the water and then it is brought in the enter that aeration process. Uniform distribution of the pollutants and the microbial degradative activity starts, and the rate of the bioremediation is speed up. Next, that is the land farming. It is simple method and low cost. In this method, excavated polluted soil is put on fixed layer support above the ground surface. तिथं जमिनीवरती पसरवून देतात. The fixed layer support above the ground surface to allow the aerobic biodegradation. It means that excavated polluted soil, whatever they entered that soil is excavated from that zone. That zone was contaminated with the help of the particular type of the pollutant. Now the area from which the soil is taken from that contaminated site, that material it is brought and it is allowed to aerobic biodegradation by the help of the microorganism. It is kept on as it is, and then the microbial activity starts. Due to that microbial activity, it can directly degrade that pollutant, and that it become it facilitates to less hazardous. It is used for remediation for the hydrocarbon polluted site. Now, advantages of the bioremediation: it is a natural process and takes less time to effort. Second, it does not use any hazardous chemicals. Third, they are eco-friendly and sustainable. And fourth, bioremediation methods are relatively easy to implement. However, some disadvantages are also there, that is in the bioremediation. First disadvantages, it is limited to biodegradable compound only. Yes, it is possible. This ex situ or in situ bioremediation, it is limited for the biodegradable compound. Then what about the non-biodegradable, like a plastic, like a metal? So these plastic and metals which will not be biodegradable, which are non-biodegradable. So these non-biodegradable material will not be degraded with the help of the ex situ and in situ bioremediation, because the microbial activity does not act on the entire such type of the plastic as well as the any type of the metal as such concern. It is only for limited for the biodegradable compound if that heavy hazardous chemical contain the carbon, and then it is possible. Second one, contaminants may be present in this as a soil, liquid, and the gases. Contaminants may be present, these contaminants should be present in the liquid form, soil form, or in the gases form. It requires a large time. And fourth one, that is the biological process is often highly specific. It is right. The biological process is specific. Whenever they enter the biological process, it is supposed to be start as a biodegradation. The biodegradation that will be present, only the biodegradable compounds are there. It means that it contains the carbon, it may be they enter the paper, it may be they enter the what we used to say that the contain waste. If that contain waste or the paper or any type of the enter that industry that has the effluent, that has the entered the hazardous chemical that contain the carbon, then they enter the bioremediation that is useful. Then the bioremediation, this process is carried out. It may be the ex situ or it may be the in situ. But limitation is that there should be the biodegradable compound. That metal, that heavy metal, or that excavated site should have the particular type of the enter that carbon. Then it is possible. Now next, that is the microbial remediation. In microbial remediation, the microorganisms are used to lower the bioavailability of the pollutant to make it less toxic to an ecosystem. It is of three types. First, biostimulation. It involves the modification of the environment to stimulate the existing bacteria capable of bioremediation. It is done by the additions of the rate limiting nutrient like phosphorus, nitrogen, oxygen, and the electron acceptor to polluted site. It is highly efficient, cost effective. The advantages: it is done by the help of the native organism, and second, it is an eco-friendly. However, some disadvantages is also there. It is the site specific and requires the keen observation. It depends on the environmental factors as such. Second, bioattenation. The principle of this method is to transfer the pollutants into the less harmful form or immobilized form. Whatever the pollutants, these pollutants are immobilized, that is arrested. These pollutants are arrested by the help of the microbial activity. It means that the pollutant remains as it is. It should not be activated again. So it is immobilized. It is done by the various chemical, physical, and biological process that reduces the mass, toxicity, volume, or the concentration of the contaminant, etc. Third one, that is the bioaugmentation. The principle of this method is to enhance the rate of degradation of complex pollutant by additions of degrading microorganism. By additions of the degradative microorganism, these microorganisms may be the bacteria. These microorganisms may be the enter the particular type of the algae. These microorganisms may be the particular type of the enter the fungi also. These microorganisms, especially the bacteria, these bacteria are enhanced. These bacteria enhances the rate of degradation of the complex compound or the complex pollutant. If the pollutant in the form of the complex form, and these complex pollutants are enhanced due to the microbial activity by the help of the entire that microorganism. The additions promote the rapid increase in the number of natural microorganisms, feeds on the contaminants, and thus enhance the degradation. It means that whatever the complex pollutants are there, whatever the content of that complex pollutants, that content of that complex pollutants that may become the food of the entire that microorganism. The microbes are collected from the remediation site, separately cultured and returned to back to the sites. Next concept is that the phytoremediation. Phytoremediation, it means that to remediate the entire that area, to remediate the particular toxic area. If that area is polluted by the help of the entire toxic metals, hazardous chemicals, or the pollutant released from the particular industry, to remediate it, to recover it, the particular type of the plants are utilized. And utilization of the plant to remediate the soil, or utilization of the plant to remediate the groundwater or water bodies or any type of the water reservoir, that is called as the phytoremediation. And these in general applied use of it, it is the general term applied to use of plant to remediate the contaminated site. The process of recovery of the hazardous substances from the soil or the groundwater contaminated with the municipal or the industrial waste by using the plant is called as the phytoremediation. It is very clear that, the entire that pollutant that may be released from the entire that municipal waste, or that may be released from the industrial waste. These municipal waste or the industrial waste, or the municipal waste, that is nothing but the sewage. What is the sewage? The water is thrown away after its use, that is called as the sewage. It may contain anything. The sewage may contain anything. Anything that directly indicate that it may contain some bacterial growth, it may contain some algal zone, it may contain some solid substances, and these solid substances that is in the form of the gravel compound. And these compound, this material that is present in the sewage that has been liberated from the entire that municipal waste. The drainage water, that is the sewage. The water which is thrown away after its use, it is simply sewage. Now the municipal sewage as well as the industrial sewage or the industrial waste, that can be remediated, that can be removed from the site. The site may be the soil, the site may be the groundwater, the site may be the entire that water body like a river, site may be the entire the water of the entire that well also. Now these can be directly recovered by the help of the entire that plantation of the particular type of the plant, and that is called as the phytoremediation. Following are the types of the phytoremediation. First, that is the phytodegradation. The name indicates that the degradation of the particular type of the toxic metals, degradation of the particular type of the hazardous chemical that is released from the entire that municipal waste or released from the entire that industrial waste, which is by the help of the entire that plant, that is called as the phytodegradation. It degrades. That plant that can directly degrades, and the degradation tends to form to less hazardous chemicals or the less hazardous complex substances of the toxic elements. Now the elimination of the organic compound by the decomposition through the plant enzymes are the product in this mechanism, the enzyme break down the contaminants to the safe compound. The particular type of the enzymes that can be liberated by the help of the entire that activity of that plant. Now the plant activity may be the vital activity, may be the entire that metabolic activity. The metabolic activity it is in the form of the photosynthesis, and that photosynthesis it take the entire that CO2 and what O2 is released. While doing this vital activity or while doing this metabolic activity, the particular enzyme that can be directly released from the plant, and that enzyme that can directly degrade. Now, the basic principle regarding the phytodegradation is that whatever the enzyme that can be directly released by the help of that plant which has been planted in the contaminated site or in the that contaminated groundwater or the contaminated the river water bodies, that can directly degrade that hazardous chemical substances or the pollutant that can be released from the that industry or the municipal waste. Now, these two types of the enzymes are there, such as the dehalogenase and the oxygenase are involved in the phytodegradation. For example, alpha-alpha grasses and the cowpea. Alpha-alpha grasses, that is the type of grasses or the grass is there. Now, this alfalfa, the plantation of this alfalfa grass that can directly degrade whatever the chemical substances, whatever the hazardous chemical substances that has been released from the industrial if it is not well disposed. Second, that is the phytoextraction. When the site is contaminated with the inorganic compound, this technique is adapted. If that site is contaminated by the inorganic compound, like a copper, boron, molybdenum, whatever it may be, they enter that inorganic compounds are there. Now, the phytoextraction reduces the soil metal concentration by cultivating the plant with the high capacity for the metal accumulation in the shoot part. Plants like a terrace, betula are used. Now, here in the phytoextraction process is that when we plant such type of the plant in the contaminated site, that site, that if it is affected by the help of the hazardous chemicals or the pollutant, that plants that will be directly reduce the soil metal concentration by cultivating the plant with a high capacity for the metal accumulation. Now, this plant will accumulate these toxic metals. This plant will store this accumulation means store. If this plant will store or this plant will accumulate some toxic substances, whatever they enter the release from that industry or whatever the municipal waste, that toxic substances that will be stored in the shoot apex. Or what is the shoot apex? It may be in the form of the leaf. Now, the leaf, that is the large area that the plant with having the large leaves, that will directly accumulate such a type of the toxic metals, and these toxic metals, that will be accumulations of this toxic metal which will be directly escape out by the help of the transpiration. Now, the transpiration, that is the evaporation of the water in the form of the vapor which is present in the leaf. It is known to us. Now, the transpiration phenomenon is that is the evaporation of the water in the form of the vapor. When these leaves that will accumulate the toxic metals in the form of the vapor at the rate of the transpiration which will be increased in the light, at that time such a type of the toxic metals vapor in the form of the vapor that will be escape out in the air, and this is nothing but the phytoextraction. The terrace and the betula, such type of the plant that can be directly utilized for the phytoextraction. Third one, that is the Rhizo filtration. This technique is used for cleaning the contaminated surface water or the wastewater by adsorption of or precipitation of the metal. Now, we are very familiar with the two phenomenon, absorption and the adsorption. The adsorption, that is in the form of the precipitation, and the absorption, that is in the form of to absorb the entire that. It is the property of the entire that substances to absorb the water. Now, the plant like sunflower, Indian mustard, rice or the spinach are utilized for the rhizofiltration. Fourth one, that is the phytostabilization. Stabilization, it means that stabilize it as it is, keep it as it is. It means that whatever the toxic metals released from the entire that contaminated site or the toxic metal which are present in that contaminated site, maybe the water bodies or maybe they enter that soil. Now, these chemical or these hazardous chemical or the toxic metals, heavy metals that can directly stabilize by the help of the entire that plant. It arrests. Stabilization means it is supposed to be arrest. Whatever the toxic substances, it is arrested by the help of this plant, and this is nothing but the phytostabilization. In this technique, the plant root and the associated microbe immobilize the contaminants. Very good phenomenon. Now, in this method, whatever the plant that contain the root, that plant has a root, and the root are present within the soil, because the root are the positively geotropic and negatively phototropic. Whatever the root zones are there, Whatever the root area, root hairs and the root zones are there, these root zones are associated with a particular type of the microbes. These root zones are associated with a particular type of the microbes, and these microorganisms may be the entire that bacteria. It may be the rhizobacterium bacteria, it may be the azotobacter, it may be the nitrogen fixing algae, it may be the any type of the entire the microorganisms which are present in the soil. Now it is very clear that the root zone and the root area which are associated with the microbes, these microbes that can directly arrest the toxic metals, and these toxic metals are not allowed to activate again, and this is nothing but the entire the phytostabilization. So that these microbes immobilize. Immobilize means arrest in the form of the prison. म्हणजे तुरुंगात थोडक्यात. It arrests as it is. Whatever the toxic metals are there within the enter that soil, these root hairs along with that microorganism that can directly arrest and immobilize, they enter the contaminants in the soil, thereby decreasing the soil and the wind erosion of the contaminations. Next one, that is the phytovolatilization. It is the uptake and the loss of the contaminants to the transpiration. This is again one phenomenon which are very associated with the phytoremediation. Phytovolatilization. Volatilization, it means that it is in the form conversion of the enter that metal in the form of the vapor. And this vaporized form of the toxic metals, vaporized form of the enter that heavy metals or the hazardous chemical, that is directly escape out by the help of the rate of transpiration. The uptake and the loss of the contaminant to the transpiration by the plant and released in the atmosphere. This is called as a phytovolatilization. The metal contaminants like the mercury, arsenic are absorbed by the help of the plant and are released in the environment. This process is called as the phytovolatilization. This is regarding the phytoremediations and the bioremediation as such concern. Okay.